So continuing with section 17.5, which was mutations, we're going to be talking about frame shift. Frame shift, as you recall, can result from either an insertion of an extra nucleotide sequence or the deletion of one. So we're going to check out what happens when we do both of those things. Once again, we have our standard wild-type DNA resulting in the standard mRNA, resulting in the normal functional polypeptide. So what happens if we were to insert an extra nucleotide? Well, the results of that could vary depending on that particular location. This particular sample is showing you if we were to insert a nucleotide at this particular location. So it looks like up above we have TAC, and then it would normally be TTC, but we insert one after the TAC. Notice that the TTC, it's still there. Alright, so that didn't go anywhere, it just got shifted one nucleotide spot this way, and everything else remains the same. What happens or could happen as a result is the change in the nucleotide into the mRNA, into the polypeptide. So if it happened to be TAC and then A right here, it would become ATT, which would translate to a stop codon. So we would have something similar to that missense one that we were looking at in the previous video there at the end. We would have our first codon, correct amino acid, and then nothing. It would stop, and this would be a nonsense result. So this would be one example of a frame shift. Notice that we didn't remove any nucleotides, we just shifted them down and we added one extra nucleotide in. So what would be the result if this didn't happen here? What if it happened down this way? Well, we would get these 3 amino acids or so, but what would happen here? Well, it depends on what would happen with the AT combination going in right around this spot. That would change the way that these codons would be read, and that would consequently change the amino acid that would be plopped in down here. So the results really, they vary quite widely. So the next type of frame shift we're going to be looking at is called deletion. And once again, wild type mRNA, polypeptide, but we're going to delete the last A, it looks like, from PHE. So AAA, we're going to delete that nucleotide sequence, and now it's going to be read as AAC instead of the AAA. What this would translate to would be a different codon. So we can see in the amino acids down here as a result of this deletion, we have some missense going on. This is leucine, and it should be phenylalanine. So once again, deletion changes the reading sequence. UUG is read instead of UUU, and as a result of this, this shifts this way, and instead of being read GGC, like we have up here, it's being read as GCU. This U, part of the stop codon, shifts down here, and actually this stop codon is no longer a stop codon, as we can see it going along this way. It would be AA, and then the next nucleotide, whatever that happens to be. So this would be a continuation of a polypeptide, and who knows what that will cause the resulting function to be. And lastly, no frame shift, but an entire amino acid is missing. This would be the result of an entire codon being completely spliced out of the DNA. So TTC is missing, which means that AAG can't be transcribed, which means that LYS, lysine, will not be in the resulting polypeptide. So the polypeptide will be lacking that particular amino acid, and that certainly would affect the functionality of this protein. So for the past little bit now, we've been talking about mutations, insertions, deletions, and those can occur spontaneously, particularly during DNA replication when we're making new cells through mitosis. Sometimes it also occurs by recombination or repairing when we need to repair. This is rather infrequent. It doesn't happen all that often. It occurs 1 in every 10 to the 10th power of nucleotides. So it happens. It's normal, but it's not, all the time happening. And the last few questions, 77 and 78, are dealing with mutagens, which are things that occur not naturally. These are outside external environmental forces that cause our DNA to change. And the two types would be physical and chemical. Physical being radiation, chemical being, chemical. An example would be taking in chemicals, smoking, first-hand, second-hand, or otherwise. The chemical type, they can have different effects on DNA. One example is called nucleotide analogs. Nucleotide analogs are things that function similarly to the normal nucleotides of the DNA, but what an analog will do is it will cause an incorrect pairing of nucleotides during the DNA replication process. So you have more and more of those errors that are made. Could those be cut out? Sure. We talked about the repair mechanisms previously, but there's going to be an incredibly more common error, so to speak, as a result of these analogs being in there. Certainly more errors leads to more mistakes that could be overlooked, causing permanent changes within our genome, within our DNA. Another example of chemical mutagens would be chemical interference with replication. So we have these chemicals that interfere with replication. How do they work? Well, they insert into the DNA entirely. These interference chemicals, they insert entirely, and this would distort the double helix as a result. Keeping in mind, one of the big things that we've been speaking about this year is that the shape of things have a direct impact on how they function. So if our DNA is distorted a little bit, its shape is changed a little bit. And a changed shape will alter how DNA ultimately can function, which certainly can be quite disastrous. This is it for 17.5. I'm going to end this video, and I'm going to start up the next one, beginning with our last section, which is 17.6.